OSU Department of Entomology, and we want to welcome you to this morning's beekeeping webinar. We have Dr. Reed Johnson with us, who's going to talk about Broodmapper, the program that he developed and uses for scientific research and citizen science. Just let me give you a little overview of the session in case you're just joining us. Please put your city and state in the chat pod so we know where everybody's from. And then we'll switch the screen over to Dr. Reed Johnson. The chat pod will be off for most of the webinar, but we'll put it back on at the end. So if you have questions, you'll have a chance at the end to type those in. And remember that the session is being recorded, so anything that's in the chat pod will become part of that recording. All right, and at this point, I'd like to turn the screen over to Dr. Reed Johnson, who is Assistant Professor here in the OSU Department of Entomology. And Reed will be telling us about Broodmapper. Reed?
Thanks, Denise. Can everybody hear me out there? Is it still good sound on this?
Sounds good on my end.
Okay, just want to make sure. Before I get into telling you about Broodmapper, I'm going to talk first about why I need to use Broodmapper and the questions that I'm trying to answer with your help using this program. So, as anybody that's kept bees knows, the varroa mite is by far the most serious pest of beekeeping, and it can be really devastating to beekeepers. It was, of course, introduced to the United States back in the mid-1980s, and it continues to be a really major problem for beekeepers throughout the country. So there are a number of ways that beekeepers can control varroa mites. There are management techniques, and there are other tools that the beekeepers can use, drone brood removal or other approaches. But in many cases, a beekeeper reaches for a chemical control. And there are a number of chemical controls available on the market for beekeepers to use. One of the first ones back in the early 1990s was a product called Apistan. And these are just these plastic strips that you hang between brood frames in the colony. And these strips contain a pesticide called talfluvalinate that is highly toxic to the mites, but is less toxic to the bees. A little bit later, of course, the mites became resistant to Apistan. So in very late 90s, early 2000s, a new product was registered called Checkmite Plus. That contains the active ingredient coumaphos, and this controlled mites for a few years. But again, the mites eventually developed resistance to it. More recently, there's been a product called fenperoximate, which is in Hivastan. This was registered for a couple years, but it's since been taken off the market, but was somewhat effective in controlling mites. Just this year in the United States, Amitraz was finally registered, and this has been a popular varroa mite control in Europe and in Canada. And there's sporadic reports of resistance, but it's not widespread yet. And then, of course, there are the natural products. Everything I've listed up to this point is a synthetic chemical. There are natural products that can control varroa mites, thymol, which is an apolite var, and apigard. And then there are the organic acids, oxalic acid and formic acid, which can be quite effective. And then there's also this hops product, Hopguard, hops beta acids. So I think some of you, if you've been watching this before, you know I've talked about the different varroa controls in previous sessions. But I think it's a really interesting topic that you can put these different compounds in the beehive, what are essentially pesticides, and you can kill the mites, and the bees are okay. And it's especially surprising because it's not a small quantity of compound that's going into these colonies. The Apistan here, you're putting 1.4 grams of this pyrethroid insecticide, basically, into your beehive when you use the Apistan product. 1.4 grams is not a small amount of pesticide, and it's really surprising that the bees can tolerate it. Similarly, none of these are small quantities. Even the quote-unquote soft compounds like the thymol, you're putting 49 grams of a chemical in there, and the bees tolerate it. And I've just always been amazed that the bees can do so well with these treatments. But a lot of it gets back to the father of toxicology, Paracelsus, who is my scientific hero. And he discovered that all things are poison and nothing is without poison. The only thing that permits something to be a poison is the dose. So to summarize, the dose makes the poison, and that's really how bees can tolerate these things in the hive. If it's the dose is low enough that it doesn't kill the bees, but the dose is high enough that it's a poison to the varroa mites. So you can make this spectrum of toxicity here. At low dose, something has no effect, and at high doses, it has a profound effect, a deadly effect. A good example in the human arena is this warfarin. So here we have warfarin bait, which is a rat poison at a high dose, but the very same molecule you can buy at the pharmacy and it may be prescribed by your doctor as a blood thinner called Coumadin, and that's at a therapeutic dose. But you'll note that exactly the same molecule is in both Coumadin and this later warfarin bait. There's no difference in the molecule between these two treatments. It's entirely the dose that's different. And obviously, your doctor wants to be very sure that you're getting a therapeutic dose and not a deadly dose. Oh my, my slides just went away. I'll wait for a moment here, but the ability of us to handle taking what's essentially rat poison and for it to be therapeutic is because we metabolize warfarin with the enzymes that exist in our liver. In fact, this is how we tolerate a lot of the toxins that we encounter in the environment. This is the reason that when you drink coffee in the morning, it doesn't kill you, because you have these enzymes in your liver that process the caffeine in that coffee so that you can tolerate caffeine, and it doesn't poison you. Same thing with warfarin, and same thing with a lot of the toxins that we encounter on a day-to-day basis. We can tolerate them because we're very good at breaking them down and metabolizing them. And the reason we can tolerate these things is because of our cytochrome P450 monooxygenases. So here's a picture of one in case you ever run into one of these enzymes. And these are important for detoxifying drugs and toxins, really in all living things. We have P450s that detoxify the toxins we encounter. Bees have P450s that detoxify the toxins they encounter, including the pesticides that they may encounter in the environment or inside the hive. So here's just an example of when you take warfarin, what would happen. It interacts with your P450, and the P450 is in black over here. You've just taken your warfarin. It interacts with the P450, gets detoxified, and is excreted. You get rid of it that way. Of course, the problem with this is that this is the reason in 4 to 6 hours you need to take more warfarin to get the same effect, because you've metabolized all of it that you took previously. So because a lot of different drugs interact with P450s, there's a lot of concern about the interactions that can occur when you take different drugs. And there's this whole school of pharmacology looking into deadly drug interactions, the interactions between different drugs that your doctor may prescribe, because of course the doctor doesn't want to kill you with drug combinations. It's not good for you. And actually the drug companies do a great deal of research into drug interactions on the human scale. So drug interactions are an issue if you take warfarin here at a therapeutic dose, but you also are taking this antibiotic Cipro, which actually affects P450s as well. You can get a deadly combination of these two compounds, which can potentially kill you. Basically what's happening here is competitive inhibition. You take your warfarin, and it interacts with the P450 and gets detoxified. And the Cipro does the same thing, but they're both competing for the same enzyme here. And as a result, you can't detoxify either one as effectively because you get kind of this backup here at the P450, and you can't detoxify either warfarin or Cipro as effectively as you could if you were just taking one of the drugs. So People don't know a lot about drug interactions in bees, even though I would argue that bees are exposed to a lot of different drugs. Many of them put there by beekeepers. We have, apistan, fenperoxamate, checkmate, who knows what a beekeeper may use in their colony. And they all are probably detoxified by the P450, and it may very well be the same P450 enzyme, and you could have a little bit of a train wreck here. So I'm especially concerned about interactions with fungicides because fungicides are designed as P450 inhibitors. So there's this whole group of fungicides called the group 3 fungicides that are designed as P450 inhibitors in fungi. This is actually how they kill fungus, is by inhibiting the P450 activity in fungus. And there's good reason to think that they can inhibit P450s in other animals as well. And here's just a small sampling of this group 3 class of fungicides. It's really a big class of fungicides. The reason I'm doing this is we want to make the bees' pharmacy guide to deadly drug interactions between the drugs they encounter in the hive and the other chemicals they may encounter outside the hive, including these fungicides. So to do this test, I did it in the lab. And this is where a lot of research on honeybees happens, because it's much easier to do these kind of experiments in the lab than it is to do it in the field. So what I did to do this lab study is I just collected frames of brood out of a colony. I allowed those adults to emerge in the incubator and allowed them to age for 3 to 4 days so that they could all be nice and their cuticle could harden. So here's a frame. This actually has most of the adult bees emerged already, but there's still a few adults emerging off of here inside the incubator. And I put those bees inside these wooden cages. We feed them sugar syrup and just let them age for 3 to 4 days so that you've got a nice aged homogeneous group of bees. There's probably about 500 bees in that box right there. Then to test for interactions between fungicides and the miticides I was just talking about, I treat them with a small dose of fungicide. And I used a microapplicator like this and just treat them on the thorax there with a very small dose of this fungicide. Allow them to recover, and then treat them with a variety of doses of taufluvalinate. This is the active ingredient in apistan. We treat them with a low dose here, medium dose, and a high dose. And then I put them in ice cream cups. So we've got everything here from a very low dose up to a very high dose of fluvalinate, or the apistan. All the bees have been treated with the same amount of the fungicide, and we're just testing to see if the presence of that fungicide makes the fluvalinate or the apistan more toxic to the bees. Then we come back 24 hours later and we count the number of dead bees in these cups. Low dose should have very few dead bees. The high dose should have many dead bees if the dose makes the poison, which is the founding idea of the whole field of toxicology. So here, just let me, this is the results from this study. So over here we have the LD50. The LD50 is the dose that you calculate that kills 50% of the bees. And if you pre-treat the bees with the control, you get an LD50 of about 12 micrograms per bee of this taufluvalinate or apistan. Now if you pre-treat the bees with a very low dose of piperonyl butoxide, you actually get a mild increase in the tolerance of taufluvalinate. But if you treat them with a large dose, you actually make the fluvalinate much more toxic. You're down to an LD50 of about 2 micrograms per bee. That's about a 5-fold increase. Now over here, these three are fungicides that are in common use on crops that bees may be exposed to, actually, because the growers can spray these fungicides during bloom, so it's entirely possible that bees would get exposed to them in relatively high concentrations. And if you give the bees a high dose of this fungicide, you can make fluvalinate, which is the active ingredient in apistan, much, much more toxic. Up to 2,000-fold more toxic if you give them a really high dose of the fungicide. I should mention that this work was just published in PLOS 1. It's PLOS 1 just a few months ago, so you can go read all about these lab-based interactions if this is something that interests you. So here's what's happening with when you give them bees this fungicide. This fungicide goes and basically stops up the bees' P450 and makes it impossible for them to metabolize fluvalinate. So basically they're getting a drug overdose. So fluvalinate used to be a very safe drug for bees. In the presence of this fungicide, it's now highly toxic because they can no longer metabolize it, and they get a drug overdose in this drug interaction. So what was therapeutic in the presence of this fungicide could become deadly, or maybe at a lower dose it could just be mildly harmful. We don't know. I mean, obviously this is all based on lab work, and we don't know what bees are actually exposed to in the field, which is the reason you need field experiments. So that was all that I showed you happens in the lab, that you get this very potent interaction between the miticide apistan and the fungicide, this whole class of fungicides. But we wanted to test this in the field to see if there's actually an interaction between these two compounds in a field setting. So here is, and actually I did this work at the University of Nebraska as part of my postdoc. This is the apiary we set up there, and we had about 30 of these small nucleus colonies set up for this experiment. And these are really small nucleus colonies, 2 frames, really about the smallest colony you can have. And we put about a pound of bees in here. And there you can see our caged queen into these just small nucleus colonies. And then we actually transferred those bees and the queen onto brand new foundation. Because one of the issues is the wax in the bee colony can accumulate these miticides over time. And so they can get a potentially significant dose of miticides just from the wax in the colony. And we wanted to get rid of any effect that might have, so we put them onto new foundation and gave them some time to draw out this foundation before we did the experiment. Obviously, we put a feeder in here filled with sugar water to help them draw out that wax. And there we are, shaking the bees into these new onto this new equipment, and we did this inside these cardboard nuke boxes, which some of you may have used. I really could not recommend them. They're pretty bad. They fall apart in the rain, but they're good disposable boxes if you want to dispose of them, as we did in this experiment because we're using various pesticides. So there we are. Our bees are installed, and they're drawing out this foundation. We give them some food to help them with that, and left them there for a week and a half or so to get them started. We filled the after a week or so, we filled the feeder here with sugar syrup containing either plain syrup, dimethoate, and dimethoate is a really nasty insecticide that definitely kills bees. We included dimethoate treatment because as a positive control, because we want to show that we can actually kill bees and cause an effect in this situation. And then we also fed them with a dose of Raleigh, which is one of these fungicides that we know can interact with P450s. And then we also either added an apistan strip between the brood frames over here, or we left it untreated. So what we have here is 5 treatments, plain syrup, dimethoate, Raleigh, apistan plus Raleigh, or apistan and plain syrup. So 5 different treatments. And then we looked for measurable effects on the colony's health. We looked obviously at colony survival. We looked at the number of dead bees, and we put dead bee traps on the front of these colonies to see if there's any obvious mortality of bees. We tracked the colony weight over the experiment. We looked at the brood area. But one of the things we really were interested in is the survival and development of brood, because this is supposed to be one of the most sensitive tests of pesticide toxicity on bees that you can measure. Now, the traditional way to look at the success and development of brood is to take an overhead projector sheet and you get a Sharpie and you lay it on top of the comb here, and you just kind of catalog the contents of each cell. And you do this for a group of 100 cells, and then you come back at some later point and see what's happened in all those cells to see if the the egg that was down here actually developed into a larva and was capped over, or if it died for some reason, or what's the status of these cells. Now this is a very effective technique and it's been used for a long time. The problem is it's very, very labor intensive, and it's kind of a big pain because inevitably you're doing this and you're sitting with the colony open, and if you're doing this in August, you've got a whole vortex of robbing bees out behind your head, and it can be quite unpleasant. So I had no intention of doing this. I wanted to use a photographic technique because, of course, we have digital cameras now, and it seems like a digital camera should be the ideal way to record the contents of cells. So I identified a 120 cell area here, and we put pins through the foundation here to mark the area and to make sure we went back to the same area each time. And then we put this is just a digital camera on top of this 1 yard length of PVC pipe, and we just put that onto that plexiglass area that I showed you a moment ago and just took a photo. And you can actually get really good photos. So hopefully you can see the eggs in the bottom of some of these cells. So there is an egg right there. There's eggs in most of these cells actually, because we had just released the queen before we started taking these photos, so she laid eggs all over the place. And then we took photos at 2-day intervals over the course of about 3 weeks. Day 4, you can see that many of these eggs have hatched, and we've got young larvae in here. Day 6, the larvae are developing nicely. Day 8, it's really amazing how fast these larvae grow up. And then by day 10, they've been capped. Notice that some of these have not made it, and the queen has relayed eggs, and you've got younger larvae. Day 12, day 14, day 16, day 18, day 20, and as you'll recall, honeybee development takes 21 days, so by day 22, those adults have emerged, and you've got a whole new cohort of young adults in the colony. And then by day 24, the nurse bees have cleaned up these cells, and the queen has actually relayed eggs in many of them. So there we've got a whole series of photos looking at the potential effects of this drug interaction in these whole colonies. Now we included about 45 colonies distributed among these 5 treatments. We have 3 images per colony because we were tracking 360 cells per colony. And we made 12 visits to each colony for taking photos, which means that we have a total of 1,620 images that need to be manually scored somehow. And that's a total of almost 200,000 images of individual cells that need to be scored, which is a real data management problem and a tremendous amount of work. So what I realized is that it's very difficult to analyze these photos just looking at them on your computer screen. It's not easy just to look at them and to try and track an individual cell over time and to figure out the fate of a particular larva. It's just not easy to do. So I began messing around with some computer programming to see if we could develop a method to more easily track the fate of these larvae. And I ended up using processing. And processing is an interesting programming language. It's based on Java, and it's meant for people in the visual arts. So it's not really, it's like a programming language for people that aren't serious programmers, which is basically me. And it turned out to be, it worked pretty well, so I worked to adapt it to a web-based version. And I learned all sorts of things about scripting and putting things up on the web. And basically we've got this program called Broodmapper, which is available on the internet, where you can go and you can look at these photos and help me score them. So that's what today's talk is about. That's what I would like to show you. So I guess this is all just a lead up to show you the justification for why you want to be involved in this project. So at this point, I am going to stop sharing this PowerPoint, and we're going to go over to a live version of Broodmapper, and hopefully this works. So hopefully you can see the Broodmapper home screen. And you can put this into your browser right now. You can go to broodmapper.com and you can probably follow along if you're interested in that. So hopefully you can see this. Denise, can you just turn on your microphone and make sure that confirm that you can actually see?
Yes, Reed, I'm seeing your browser and the purple bars with rank name frames.
Excellent, thank you. Okay, so it's not a real classy front page because it's kind of still in development, and I didn't put much effort into the home page here, but underneath it all works. So on the front here we've got the really the high scores for this program, and you can see people that have scored almost all of the frames that have been available. Bob, he was actually an employee at the University of Nebraska, and he scored all of them. Number 2 down here is actually my mother. She has also scored almost all the frames. And you can work your way up here. So if you score a lot this week, you can actually get into the top Broodmappers this week. Anyway, well, let's go see what this is all about. So if you log in, I'm sorry, first thing you want to do is sign up with a new user account. And I am going to make just a new user account so that you can see the tutorial that it's going to make you go through. And you can make just a junk password, and you can provide an email address if you like. Email address is not required. And just sign up. Okay. So then you just log in, just like any website you go to. And the first thing it will do is click here, and it will take you to a welcome screen. So the first thing that Broodmapper makes you do is go through a series of training frames in order to help you learn how to interact with the program and how to score cells effectively. So welcome to Broodmapper. Thanks for joining the hive mind to help honeybee research. So to go to the next frame, you always want to click on the next button over here on the right in this Broodmapper toolbar. It will tell you that stages have been saved. That doesn't really mean much right now, but it will in a minute when we start actually scoring some frames. So I'm just going to resize the window here so we can see everything. This first tutorial screen just gives you some idea about how you can navigate within the program here. You can adjust your view by clicking on the arrow keys over in the toolbar over here, and you just click on those, and it goes up, left, right, and down, and up, down, right, and left. You can zoom in with the buttons over here. Now you can do that with the mouse. You can also do this on the keyboard. The arrow keys, and I'm pressing the arrow keys on my keyboard, will move the screen up, right, down, and left. And the page up and page down keys will zoom in and out. One other way to move around is with the mouse, and you can click and drag the image to move it around like I'm doing now, and you can use the scroll button or the scroll wheel on your mouse to zoom in and zoom out. Now I took a lot of effort to make it easy to move around in here because that's a lot of what you do in analyzing these things is you need to zoom in and see what's in these cells and then score it. So it's worth taking a moment to get familiar with how you can move around in the program. Then you click Next. Stages are saved. And here we actually have to score some images. And this is just a little tutorial in the stages of bee development. And I've managed to find a photo that has all the different stages of bee development all in one photo. So we're going to start up here with this egg that's been identified in green. And I'm going to zoom in on these two cells that are in circles, in the black circles up here. And I'm just zooming in with the mouse scroll wheel. And you can see that there's definitely an egg in this circle on the left, and there's a young larva in the image on the right. Now to score these cells, what you would do is you would go over here to the egg button, and that turns on egg mode. And then you come back here and just click anywhere inside this circle, and it will make this little kind of cartoon egg, and that's how you know that you have manually scored the contents of that cell. Now this next one has a young larva in it. We're going to go over here and click on the young larva button, and then click on anywhere inside this circle and score that as a young larva. Of course, after the egg hatches and the larva grows into a middle larva, and the middle larva is here in the middle of the frame, we're going to score that by clicking on the middle larva button and the larva here. Next, it develops into an old larva. Click on the old larva button and the cell here. Finally, that larva gets capped, and we click on the capped button over here, and then click inside the cell over here on cap to score that as being capped. And then if you're lucky, you will see a new young adult bee emerging, and we actually have a code for that as well with this emerging button here. So we have scored all of the cells that were identified on this frame, so we can click Next to go to the next training frame. It will indicate the stages are saved. You click OK. So now let's score these again. Got the egg, young larva, middle larva, old larva, capped, and emerging. Now, at this point, if you had not scored one of these, it would not let you proceed. Now, it's kind of a judgment call when you're looking at the different stages of larva. What kind of larva are you really looking at? Now, up here in the upper right corner of this frame, we've got what I would call old larva. An old larva really fills the entire cell. So we're going to score all these up here as old larvae. Now, if you have any question about what an old larva looks like, you'll notice that there are these question marks next to the buttons here. If you just press on this question mark, it will bring up a screen with examples of what I would consider old larvae to look like. And as you can see, all these examples really fill the entire cell. There are examples available for all the different kinds of cells that you will be asked to score. These in the middle here I would call young larvae, or middle larvae, and we'll go ahead and score those as middle larvae. And a middle larva, I say, is shaped like a C. They're bigger than small larvae, but do not fill the entire cell. And then we have young larvae, which are shaped more like a parenthesis, and we'll go ahead and score these young larvae here. Okay, we scored everything on this frame, so we'll go ahead and click on Next. All the stages are saved, so we can go on. Now, of course, not all the cells in a bee colony are filled with larvae. There are also nectar and pollen in cells. And that's what we've got on this frame. The shiny stuff here is nectar. We've got nectar here and here, here and here and here. You might occasionally see honey, and that will be capped over with a wax capping. These were all very young colonies, so they really didn't have a chance to make much honey yet. So it's going to be very rare that you see honey in any of these photos. We do see some pollen, though. The bees were collecting pollen quite effectively. And you notice it's all this orange pollen in these photos. You just score that with the pollen icon. And then click Next. Now, obviously, sometimes you will see different colors of pollen. It's not all orange. And then, of course, because this is a pesticide experiment, sometimes there are dead bees that you'll see occasionally. And we do have a scoring for dead over here. And these are pretty clearly dead larvae. They don't look like any of the things that you've seen before. They look kind of like slumped in some cases rotting larvae in the bottom of these cells, which is, they can sometimes be really difficult to figure out exactly what is in those cells because they do look so degraded. So we score those 2 cells as dead and click Next. Now, one of the problems with doing this with a digital camera is sometimes we don't get good photos. Sometimes the photos are out of focus, or sometimes that plexiglass frame that we use to mark off the cells gets in the way. And that's what you see here, is sometimes these empty cells are, you can't really see to the bottom of the cells because the plexiglass is in the way. And I'm sorry, I just filled these with eggs. If you ever fill something and you want to unscore it, you can click the clear button and you can clear it out again. So what we're going to do is we're going to zoom in and we're going to really try to see what's in the bottom of these cells here, if we can see anything at all. So this one on the left is clearly obscured by the plexiglass. So we're going to go ahead and click that as unknown because we really can't know what's in there. And all of these are really unknown because I misaligned the plexiglass and there's just no way to know what's in there. Now down here we can pretty clearly see an egg, so I am going to score that as the egg. This one, however, does not have an egg in it. That one is empty, so I'm going to score that as empty. And this one has no egg either, so that is an empty cell as well. Zoom back out, make sure I got all the cells that were highlighted. And then go ahead and click Next. Oh, I forgot to mention on this frame, there is an invert function. And sometimes that can make the eggs easier to see if you invert the colors on the frame. And hopefully this is working. Some computers have more problems with the invert than others because it takes a little bit of processing power to invert the image. But sometimes this can make the eggs much more visible if you invert the colors. Okay, so we'll go on to Next here. So now we got a little quiz for you, and you can just go through and make sure that you can actually, I put some deliberately challenging cells on here to give you a little bit of a challenge. So I'm just going to go ahead and complete these here, if I can. And so this top left one here, you may not be able to see it. I'm going to invert it, but there is actually an egg in this cell here, and you can hopefully see my cursor. It's right there. That one contains an egg. Turn off the invert. This one is empty, though. I don't see any sign of an egg there. There's an egg in this one, and an egg here. But this one is empty. I'm sorry, empty. As is this one. That looks like a little bit of maybe leftover pollen, but I would call that empty. This one contains an egg. Empty. Empty. Egg. Empty. And egg. Okay. So there, you can see that the eggs are really the most challenging cell type to find, because sometimes they can be very, very tricky. But just do your best. The different stages of larvae can also be a tricky thing. And this is kind of more of a judgment call, and you can just do your best. This is very clearly an old larva, a young larva, and a young larva, a middle larva, another middle larva, an old larva, let's say that it's a young larva, middle, 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 young, middle, young, young, old, and this one, that larva is not looking healthy at all. I'm going to score that one dead. Let's see, did I get it right? Now I should say that if you try to just go through this without scoring them and you just try to hit the next button, it will tell you that you have not scored some of the cells, and it is going to highlight those in green, which can be a problem if you're trying to see these. So once they're highlighted in green, you can just go down here to clear and clear out that green color so that you can see them and score them, and hopefully it will allow you to go on. So are these capped brood or pollen that we're looking at here? This is pollen up here. This is all brown pollen, which is the easiest to confuse, brown or orange. Those are all pollen. And then these ones with the kind of smooth capping, or this one is just in the process of being capped, those are capped cells. And now it's going to let me proceed. The other thing that may be confusing is the difference between young larvae and nectar. And the reason this is confusing is that that jelly that's at the bottom with the young larva reflects the flash from the camera in much the same way that the flash is reflected in the nectar cells. I should also say that sometimes you can't actually see the young larva, but you can see the jelly that the young larva is floating in. Notice that it's got this milky color, whereas the nectar is pretty much completely clear. So we've got young larvae here, 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 and here, and the rest of these are nectar. And here's a real grab bag of weird things that you might see. Some of these you just have to score as unknown because it's impossible to know what's in the cell. This one, the plexiglass is blocking it. This one, there's a nurse bee feeding something down there, but you can't see it. More plexiglass. This one, there's plexiglass blocking it, but you can still see the egg in the bottom there. This one, there's a bee covering it, but I think I see an old larva down underneath that bee. This one looks like a middle larva, an old larva, and this looks like some pollen, even though it's still blocked somewhat by the plexiglass. This one, I can't see what's down there, so I'm going to mark that as unknown. This one has an egg. This one is unknown. And this one looks like a capped cell to me. And then finally, here's your final exam. And you're going to be asked to correctly identify all the cells that are in this arena here. And I'm not going to go through that. I'm going to actually log out. And I would ask you to go ahead and complete this. I'm going to log out and show you what cells look like when you actually begin the process of scoring. After you complete that tutorial, you'll be taken to a set of real cells that look a lot like this. Now you notice here we have this 120 cell arena, and you have got 120 cells that you can potentially score. Now I just screwed up and I scored a bunch of things as cells, as eggs when they're not really, so I'm going to go down here to the clear button and clear these out. You zoom in. Now when you're moving things with the mouse, you want to click and drag on the area between the cells. You can't actually click on the cell itself because that will score the cell. But you can really zoom in here and see what's in these cells and begin to score them. And that's really what I'm asking you to do. So what you'll do is you will see a series of 12 photos, all with 120 cells, and you're gonna be following those same 120 cells through development. So you're actually going to see individual eggs grow up into larvae. Those larvae will get capped over, and then hopefully you'll see the adults emerge. So you're going to really see the entire story of particular cells as they complete development over the 21 days of the experiment, which is really an exciting thing to see. I really got, this is a view of the beehive that you just can't get if you're actually out there doing beekeeping, because you just can't get this close. And you can't take your time like this to see what's going on in all the cells. And this is the reason that I really, apart from helping me score these photos, this is really an interesting thing, I think, for beekeepers to look at, because you can really hone your ability to assess different life stages of the brood, and you can really get a view of these cells like you would never be able to get otherwise. So I'd encourage you to go ahead and sign up, work through the tutorial, and work through at least one set of these cells. Obviously, there are a whole large number of photos that you can work through. Now, you'll want to remember that some of these bee colonies have been essentially poisoned, and you might see dead bees, and you might see some really weird things going on in there that you would not see in a healthy colony. But it's important to go ahead and score that, and to just score as best you can. Now, I will say that if you ever see a problem that you can't, that you want to report, there is a report button up here that will allow you to report a problem with a particular image, and you can just go ahead and report whatever the problem is. That there's a usually the problem might be that the cells don't line up with the photo that's underneath. That's a fairly common problem, and something I'd definitely like to know about, but you may find other problems that you want to tell me about, and this is your way to do that. Also, if you ever want to go back to any of those tutorial frames, you can click on the tutorial button here on top, and it will take you back to any of the frames that we just completed, including the summary frame, which contains a whole list of keyboard shortcuts that you can, rather than coming over here to the right of the screen and clicking on the different buttons, you can just click the E key if you want to score something as an egg, the 1 key if you want to score something as a young larva, or the space bar if you want to clear a cell. So these are keyboard shortcuts that I made to help it make it easier to score the different cells. You can go back and go to your frame again. So I'm continuing to develop this program. I'm hoping to take some more photos to put on Broodmapper this summer in an experiment we're designing. So if you can get on here and try this out, and hopefully later this summer we'll actually have some more cells for you to take a look at. Now, before we open it up for questions, I'm just going to go back to Carmen Connect, and I just want to show you some of the oddities. You take pictures of 200,000 cells, and let me tell you, you see some weird things down there. I'm just going to go back to this Broodmapper document and share this again. And I want to show you some of the craziest things that you might see down there, just to show you what you may be seeing, and you might get lucky enough to see something crazy like this in your own cells as you're scoring them. We'll see if this can load again. I should say that the biggest challenge in doing this is getting good photos. And the photos that we've got, I think, are pretty good, but some of them aren't that good, and you may not be able to see the bottom, and just please do the best you can with those. So I'm just working through the demo here. And before I go on, I'd just like to thank all the people that helped make this possible. This, everything that I've shown you to this point was all funded through the United States Department of Agriculture Managed Pollinator CAP grant. And this was the big grant that was awarded in response to colony collapse disorder announcement back in 2007. And it's really a multi-institutional project that involved, I think, 12 different universities. And my postdoc was funded by this Managed Pollinator CAP project. And this is one of the products of that project. So oddities that you might see down here. Here is a particularly interesting oddity that you may run across. And I don't know if anybody knows what that is that you're looking at there. You're actually looking at a bee's posterior. This is the sting right here. That bee's head is down in that cell. And I don't know if she's eating something or if she's feeding a larva, but you can't see the contents of that cell because you're looking at a bee's behind, basically. This is an interesting thing that you might see. Those are actually 2 pollen balls that have just been unloaded by a forager bee and have been dropped off in this pollen cell, and they haven't been processed into bee bread yet. But it's interesting to see the pollen ball still sitting there on the surface. This is actually pretty rare to see this. The bees must process these pollen balls into bee bread quite rapidly because it's pretty rare to see the balls just sitting there. Now, some of you may think that this is a laying worker here, because laying workers will commonly lay multiple eggs per cell. However, you'll notice that these eggs are all on the bottom of the cell. Laying workers would lay eggs on the sides of the cell. So this is actually a queen that has laid multiple eggs in these cells. And this is probably because in the process of doing this experiment, we caged the queen for about a week here, and I guess she kind of got backed up with eggs. And once we let her out, boy, she just laid a lot of eggs. 4 eggs in one cell laid by the queen is the most that I have ever seen, but kind of an oddity. What's here? We've got twins. This is, if there's multiple eggs in a cell, sometimes those will develop into multiple larvae in a cell, which is really another kind of amazing thing to see. And these are it's not too uncommon to see small larvae sharing a cell, but to see larvae of this size sharing a cell is pretty rare. And this is by far the weirdest thing that we saw. Those are 2 fly pupae that are on top of a pollen cell in this colony. And I'm pretty sure those are fly pupae. What they're doing in this bee colony, I don't know. Some of you may have heard about these zombie bees. It's a phorid fly that is a parasitoid of bees and will get into honeybee colonies and parasitize the honeybees. And it's possible that these are pupae from that parasitoid fly. They're about the right size, so it's possible that this is one of those parasitoids. Or who knows, it could be any other sort of fly out there. There's lots and lots of flies out there in the world. And I have, the picture unfortunately is not good enough to actually get a good ID. It's hard to ID anything from a pupa anyway. But they certainly look like fly pupae, and it would be really interesting to know what kind of fly eventually emerged from that pupa. But so there you go. That's the freak show. Among these 200,000 cells, these are the craziest things we saw. There may be other crazy things going on there, and I would encourage you to go ahead and hit the report button if there's anything really weird that you see down there. So please go ahead and sign up and complete the tutorial and work through some of these frames, either for your own enjoyment, and I hope you do find it somewhat enjoyable, and to help out with the research that we're doing on the effects of pesticides and pesticide combinations in whole bee colonies. So at this point, I'd be happy to take any questions that you might have.
Reed, this is Denise. Can you talk about how the data comes back to you and how you use it?
So what we do is we make a survival curve for each of these cells to see if the individual egg that was laid in that cell survives to adulthood. And so we're looking to compare survival among the different treatments between the different cells. So in the end, we have, I think, about 15,000 eggs that were laid and that go through to potentially to adulthood. And that's what we're comparing, is the survival of these eggs through development to adulthood, and that's where we're looking for the effects of pesticides or pesticide combinations on the bees. But all of that data is stored automatically. There's nothing you need to do. It all goes into a database on the web server, and then I can download that and do my statistical analysis on it.
Sandy asked, how often do you get pictures to evaluate? So I'm guessing Sandy's wondering every time, I'm sorry, Sandy, he or she logs in, then you get more images to go through?
You do. It will just keep giving you new images until you log out, basically. There are about 1,600 images, so you could sit there for a long, long time and you would never see a repeat. It's set up so that it will not give you repeats. You'll always get a picture that you haven't seen before.
Earlier, Reed, when you were talking about your research, Swarmcatcher put a question in and wondered if you feel the research would have changed if your frames were given honey instead of sugar solution.
It's possible. The problem is it's hard to. We could have given them honey, but it's just much easier and it's much more standardized to just give them sugar syrup. Who knows? They might have changed, but that's another experiment for another day that could be done to look at that interaction between the honey and the fungicide and the acaricide. But then you're looking at a three-way interaction, which will be a much larger experiment. So, I mean, I think the place to start would be to look at this in the lab to see if you can affect the toxicity of taufluvalinate in the lab with the honey and the fungicide, and then if you see an effect there, then you would take it to the field and actually feed field colonies honey in an experiment like we did here.
There's a question about where the results are published.
The lab results are published in PLOS One, and if you just search my name, Reed Johnson, in PLOS One, you will get a link to that entire lab-based study. We have not published the results of this field-based study yet because we are still scoring the photos. So go ahead and score the photos and help out.
And Anne wonders if it's possible to see a lethal interaction at lower doses over a longer period of time.
It's possible. We didn't look at that. I mean, of course, everyone's concerned about sublethal doses. I don't know that we would, because the toxicity for fluvalinate, it either kills them or it doesn't, but there may be a longer-term effect that you could detect there as well. And that would be an interesting experiment to do in the lab, is to run these experiments out to a week or something to see if you can see an effect on bees' longevity.
Oh, great, Carolyn, thanks. Carolyn put the reference there for that PLOS One article. Thanks, Carolyn. Bruce wondered if the cells are scored by multiple scorers.
Yes, they are, and that's actually an important thing. So you should not feel like the success of this research is entirely based on your scoring. So just do the best you can. Multiple people will score the same image, and what we do is we take the consensus among all the different scorers and assume that that is the correct scoring for that particular cell. So we're really looking to crowdsource this. If everybody just looks at a few of these photos, you can really make a major contribution. It's not a huge time commitment I'm looking for. Just go in there and score a few and do your best. And by having many people work on this, we can effectively get through this big stack of photos.
It's great, Reed. Thank you. And I think it's a really neat way to bring folks who aren't beekeepers, as well as folks who are beekeepers, in to help with this citizen science. It's a really cool idea. Folks, if you could put a thank you for Reed down in the chat pod, I'm sure he would appreciate that. Many of you found the 2 poll questions we have open. If you could just let us know how long you've been a beekeeper, if you are one, and your impression of today's session. And I also want to remind you that next month we're going to have Kim Flodham with talking about setting up the honey house. He's the editor of Bee Culture magazine, and he'll be here on June 19th at 9 a.m. So thanks, everybody. Thanks for joining us. I'll have the recording posted later this afternoon. Thank you, Reed, for your time.
Thanks a lot for everybody. Thanks for helping out.